Our second speaker is Rohan Khadilkar, and he's going to tell us about modulation of occluding junctions alters the hematopoietic stem cell microenvironment to trigger immune activation in the fly.
Good evening, everyone. I'm Rohan. I'm a postdoc at the Guyton and Zap lab at University of British Columbia. And I would like to thank the organizers for giving me an opportunity to give a talk today. And today I'm going to tell you a story about the role of occluding junctions in the hematopoietic stem cell microenvironment in Drosophila. One of the main interests of our lab is in understanding how stem cell fate decisions are made, wherein a balance is maintained between the stem cells and the differentiated cells in order to maintain tissue homeostasis, failure of which either leads to a cancerous or a tissue degeneration scenario. One of the primary mechanisms by which this can be achieved is by the presence of a physical microenvironment called as the stem cell niche, which produces instructive signals that could be either required for the self-renewal of stem cells or for priming them towards differentiation. Basically, there could be two mechanisms at play. The first being the exit from the niche, wherein the stem cells that are meant to self-renew reside at the niche, whereas the ones that are meant to self-differentiate gradually make an exit away from the niche. And the second mechanism could be by the formation of an isolating barrier, which provides a physical microenvironment in order to provide a favorable signaling milieu for the self-renewal of stem cells. Now, one of the most important candidate class of molecules that is required for formation of such an isolating barrier are occluding junctions. And surprisingly, occluding junctions have been shown to be upregulated in a number of vertebrate stem cell niches. So we were really curious to understand if this is also true in the case of the Drosophila stem cell systems. Now, occluding junctions are called as septate junctions in the case of invertebrates of lice, and they are located basally to the adherence junctions, whereas their functional counterparts, tight junctions, are located apically to the adherence junctions. And their primary function is to regulate the paracellular diffusion of molecules. One of the key components that is required for the formation of septate junctions is neurexin 4, which is the core transmembrane component, and its intracellular binding partner is coracle. And these are the two primary molecules that we have analyzed in this particular study. And the system that we have focused on is the hematopoietic system in Drosophila, particularly the larval lymph gland, which is the hematopoietic organ. And shown on the left is the schematic of the primary lymph gland lobe, which is the main site of hematopoiesis in the larvae. And on the right is the image of the same. The primary lymph gland lobe is divided into three different zones: PSC, which is the posterior signaling center, which acts as a stem cell niche; medullary zone, which houses the prohemocytes or the hematopoietic progenitors; and the cortical zone, which houses the differentiated hemocytes. Now, hematopoiesis in Drosophila leads to the formation of three different differentiated hemocytes: plasmatocytes, crystal cells, and lamellocytes, and their functions range from the formation of antimicrobial peptides to the phagocytosis of bacteria. So we started off by looking at the expression of septate junction components in the hematopoietic stem cell niche. And what we find is that indeed, my slides seem to be messed up. The septate junction molecules are specifically enriched at the hematopoietic stem cell niche, but they are uniformly expressed in the entire primary lymph gland lobe. And if you look at the electron micrographs of the niche cells, you find the presence of beautiful pleated septate junctions between the niche cells. Since the primary function of these molecules is in regulating the paracellular diffusion of molecules, we looked at the permeability of the tissue using a dye assay. We devised a dual dye assay wherein we looked at the permeability of the tissue by differentially labeling the different molecular weight dextrans as fluorescent conjugates. And when you look at the wild type tissue, what you see is that the 40 kilodalton dextran can label all the primary lobe lymph gland hemocytes, whereas the 70 kilodalton dextran has a specific exclusion in the niche cells. So we see a molecular exclusion cutoff between 40 kilodalton and 70 kilodalton at the niche. So since we know that the septate junction molecules are specifically enriched at the niche and they form a permeability barrier at the niche, we were interested to see if they have a niche-specific role. We then depleted the septate junction components in the niche using a niche-specific Galfo driver. And what we see is that now the 70 kilodalton dextran is able to access the niche cells, shown here. And if you quantitate and look at the colocalization coefficient of the two dyes, you see that there is a higher colocalization coefficient upon the knockdown of septate junction components in the niche as compared to the controls. Now, the septate junction knockdown niche lymph glands also lead to a number of phenotypes, the most striking being the loss of blood cell homeostasis, which is shown in a number of other mutants and molecular mutants as well. So we see that the blood cell homeostasis is lost and there is increased amounts of precocious differentiation. So we were really interested to know if septate junctions are acting specifically at the niche in regulating the blood cell homeostasis. So we started looking at a number of mutants of different septate junction molecules and Drosophila claudens, and what we found is that there is increased amounts of differentiation in these mutants. So we tried to see if restoring the function of these septate junction components specifically in the niche is able to rescue the mutants. And when we did that, what we found is that the septate junction component overexpression, specifically in the niche, in the genetic background of the septate junction mutant alleles, is able to rescue the primary lymph gland lobe phenotypes. And since what is already known in the field is that systemic signals also influence lymph gland hematopoiesis. So we wanted to see if there is any contribution of septate junctions in regulating lymph gland hematopoiesis in a systemic manner. So we perturbed the septate junction components in a number of tissues, including the gut where it primarily functions. And when we did that, what we found is that there is no effect on lymph gland hematopoiesis upon knockdown of septate junction components in the gut. So we wanted to know if the breakdown in the permeability barrier is causing any localized changes in and around the niche cells. So we looked at a number of signaling pathways, one of them being BMP, which is primarily active at the niche cells, and we looked at the DAT-GFP reporter to look at BMP signaling. And what we find is that in the wild-type situation where DAT-GFP is primarily active at the niche, in the case of the septate junction depletion in the niche, the DAT-GFP signal gets further extended on away from the niche, which shows that indeed there is localized perturbation of the signaling microenvironment in and around the niche cells. So what we knew so far is that there is loss of septate junctions from the cell membrane that breaks down the barrier and brings about a change in the signaling microenvironment that leads to the increased production of these differentiated hemocytes, which primarily act as immune cells. So we asked a very simple question. If the breakdown of the permeability barrier is a part of the natural immune response of the flies. To test this, we infected the Drosophila larvae with either gram-positive and gram-negative bacteria, and we looked at the expression of the septate junction components in the lymph gland PSC. When we did that, what we find is that the enrichment that you see in the sucrose control is now downregulated upon either gram-positive or gram-negative infection. This also translates into the permeability assay, wherein if you look at the wild type where the dye is not able to access the niche cells, when you infect these larvae with either gram-positive or gram-negative, the dye is now freely able to access the niche cells, the quantitation for which is shown here. And this very well also correlates in the phenotype that we see with the septate junction depletion in the niche, wherein there is increased amounts of plasmatocyte production upon infection. So the barrier breakdown is indeed modulating the environment and coaxing the prohemocytes towards differentiation. So we then asked if barrier breakdown is acting as a boon or a bane for the adult flies. So we basically, what we did is we did a survival assay upon infection, wherein we took the flies having niche-specific depletion of septate junction components and compared it to the wild type. And what we see is that flies having septate junction depletion in the niche are better off as compared to the controls in both gram-positive as well as gram-negative infection. So indeed, niche-specific depletion is giving them an immune protective ability against an infection. So we now wanted to know what is the connecting link between infection and loss of septate junction components, which is leading to the barrier breakdown. To test that, what was well established in the field is that gram-positive or gram-negative infection specifically triggers either the Toll pathway or the IMD pathway. And so we basically wanted to genetically mimic an infection by immune activation. So we did Toll and Imd activation in 3 different ways. We did a localized activation only at the niche. We looked at the constitutively active mutants of Toll and Imd, and we also did a systemic activation of Toll and Imd in the gut. And in all the 3 cases, what we see is that there is a barrier breakdown at the niche, which you wouldn't see in a wild-type situation. So infection is inducing the Toll and Imd response, which is leading to the loss of septate junction components from the cell membrane, leading to a change in the microenvironment, leading to the increased production of immune cells. So then the most exciting thing to do was to see if barrier breakdown in the immune deficient mutants of Toll and Imd is able to rescue these immune deficient alleles. So we created double mutants of immune deficient alleles of Toll and Imd with the septate junction mutant alleles, and we did a survival assay upon infection. And when you look at the survival assay, what you see is that as compared to the immune deficient alleles which perish soon after infection, the double mutants show a partial rescue, which shows that partial loss of septate junction is able to rescue the survival of the immune deficient alleles. So then the obvious thing to ask was whether septate junction component depletion in the niche is triggering the cellular or the humoral immune response or both. So we started off by looking at the humoral immune response, and we specifically looked at the Toll and the Imd reporter in the fat body, which is the drosomycin and the dipterosin reporter. And we looked at the GFP expression in the conditions of niche-specific depletion of septate junction components and compared it to the control. When you look at the humoral immune response, we see no endogenous activation of the GFP reporter in the situation of septate junction component depletion in the niche. The reporter gets activated only upon infection, and there is no difference between the control and the knockdown. So we indeed see that there is no difference in the activation of the humoral response. So we then looked at the cellular immune response, which is manifested mainly by the hemocytes in circulation. And when you look at the circulating hemocytes in the early pupal stages, where the lymph gland would burst open and would release all its terminally differentiated hemocytes into circulation, what you see in the conditions of septate junction depletion in the niche is that there are increased amounts of these differentiated plasmatocytes as compared to the control, which is also shown here. So indeed what is happening is septate junction component depletion in the niche, or the downregulation in the niche, is breaching the permeability barrier at the niche, leading to the formation of increased amounts of these cells, which are gradually released into circulation. So there is increased circulating hemocyte load, which is mounting a robust cellular immune response. So to summarize, what I showed you today is that in a wild type or a normal physiological scenario, septate junction components are enriched at the niche and they form a permeability barrier at the niche, wherein they regulate the flow of these differentiation factors across the niche cells into the other parts of the lymph gland, preventing precocious differentiation of these cells. Whereas in an infection scenario, what happens is the septate junction components get downregulated, leading to a breach in the permeability barrier, which would then trigger differentiation of the prohemocytes. And the way we conclude is that stem cell niche, by employing mechanisms like presence of a permeability barrier, can now act as a molecular switch between the normal physiological and the disease scenario. And I would finally like to acknowledge my supervisor, Guy Tenenzap, and my wonderful lab members. Thank you all for listening. Happy to take questions.
We have time for one question.
So, when you overexpress coracle and you then immune challenge your larvae, can you prevent activation and precocious?
Yeah, we tried to do that, and we do see that the permeability barrier is not breached, even upon infection, and there is no induction of differentiation.
So this is only happening with a natural differentiation at the end of larval life?
Yes, yeah.
I see.
Yeah, in fact, it completely suppresses the plasmatocyte lineage.
Does it then suppress when you overexpress in the posterior signaling center? Does your lymph gland not come apart and not differentiate?
Sorry, I didn't get your question.
So does it suppress the natural differentiation?
Exactly, yeah.
So this is happening?
Yeah.
But you haven't shown it, but that...
No, I haven't shown it today,
okay.
No.
More question? Anna?
Since the main function of the PSC is in fact to control the cellular immune response to wasp parasitism, did you look in wasp infection conditions if you change the permeability barrier of the PSC? Because it's a typical situation we should look at that.
Yeah, definitely, but we have not looked at wasp infestation at all. We have just focused on bacterial infection for now. Thank you.
Great. Thank you, Ron.
Thanks.
Our next speaker